A blue-gray fish emerges from volcanic darkness, moving fins like paired limbs through a submerged cavern. For modern science, the coelacanth once existed only as fossils from a vanished branch of history. Then, a specimen reached a South African dock in 1938, overturning that assumption. The mystery is not how a fossil returned, because no extinct animal came back. If you follow the evidence, a surviving lineage had simply remained outside scientific recognition. Concealment arose from habitat, geography, fishing history, and the limits of human sampling. Local fishers encountered these animals before zoology formally described the living species. Scientific absence, therefore, reflected incomplete knowledge, not an empty ocean. The investigation begins with one damaged catch and moves into steep nocturnal habitat. It asks how a large fish avoided documentation while people worked nearby seas. The answer requires separating evolutionary survival from the much shorter history of human discovery. Coelacanth relatives were diverse in the fossil record, but most lineages ended long ago. Living coelacanths are surviving branches, not unchanged replicas of every fossil ancestor. Their story joins persistence in deep refuges with the narrow reach of early marine science. Beyond the harbor, darkness kept an ordinary animal extraordinary to distant observers. Museum curator Marjorie Courtney-Latimer recognized unusual features in the 1938 fish from East London. Ichthyologist J.L.B. Smith later identified it as a living coelacanth and named Latimeria chalumnae. A second specimen obtained near the Comoros in 1952 confirmed that the first was not an isolated impossibility. These catches provided physical bodies, enabling anatomy to replace speculation. Fleshy paired fins, a distinctive tail, and an intracranial joint helped establish its identity. The fish also possesses a notochord filled with fluid rather than a typical ossified vertebral column. Such traits connect living Latimeria with coelacanth evolutionary history, while many details remain specialized. The verified animal inhabits deep rocky slopes and caves in tropical marine environments. Daylight observations from submersibles show individuals sheltering in cavities, sometimes gathered loosely together. Darkness is the cue, and coelacanths leave shelters to drift and forage along steep terrain. Their flexible fins stabilize slow movement, conserving energy while currents carry them near possible prey. This cue-action adaptation sequence makes nocturnal habitat both feeding ground and protective screen. At dawn, individuals return toward caves, reducing exposure in open water. When you picture their refuge, imagine broken lava slopes rather than an unreachable abyss. Depth, rough bottom, and limited daylight made conventional nets and early observation unusually ineffective. Coelacanths do not commonly inhabit shallow coastal flats where naturalists historically collected many fishes. Their rocky habitat can damage trawls, discouraging systematic sampling by large bottom fishing vessels. Traditional deep handlines could reach them, but catches were uncommon and geographically concentrated. A caught animal might be known locally without entering a museum connected to international taxonomy. Recognition requires access, preservation, expertise, communication, and the luck of examining the right specimen. In 1938, Those conditions finally aligned around a single harbor catch. Later expeditions moved from accidental specimens toward observation of living behavior. Submersibles documented coelacanths, maintaining head-down postures and maneuvering precisely near rocky walls. Their paired fins alternate in coordinated patterns resembling tetrapod limb sequences superficially. That resemblance does not mean coelacanths walk on the seabed or directly reenact land vertebrate evolution. Direct observations show hovering, drifting, and controlled swimming rather than underwater walking. Currents shift around the slope, prompting fin adjustments that hold position with limited muscular effort. Energy conservation follows, suiting a large predator in relatively low productivity deep habitat. Diet evidence includes fishes and cephalopods, inferred mainly from stomach contents and anatomical studies. Actual strikes are difficult to witness, so precise hunting tactics remain incompletely documented. An electrosensory rostral organ may help locate prey, especially when vision is limited. That functional interpretation is mainstream, but its exact role during natural attacks remains under study. A second living species, Latimeria menadoensis, was scientifically described from Indonesia in 1999. Its recognition widened the known geography and exposed another gap in previous sampling. Genetic and anatomical evidence distinguishes the Indonesian species from Latimeria calumnae. Neither discovery implies coelacanths occur everywhere between their separated known populations. Verified populations are associated with particular slopes, islands, and suitable daytime shelters. Environmental limits appear important because resting caves must remain cool and sufficiently oxygenated. Temperature cues influence depth choice as individuals seek water within their physiological tolerance. Movement into suitable layers prevents metabolic stress while preserving access to nocturnal foraging habitat. This behavior helps explain persistence within narrow environmental corridors. It also means apparently nearby coastline may offer no usable refuge. Reproduction deepens the pattern of slow, hidden existence. Coelacanths live young after internal development, as documented from examined females and embryos. Evidence indicates long gestation and low reproductive output, though exact natural intervals remain uncertain. No scientist has comprehensively observed courtship or birth in the wild. Claims about elaborate mating rituals would therefore exceed direct evidence. Slow reproduction may have supported stable life in persistent habitat, yet it restricts recovery from losses. Their survival outside scientific knowledge was real, but it never meant safety from environmental change. Coelacanths occupy food webs as large, slow-moving nocturnal predators. Leaving a cave after dark brings them near fishes and cephalopods distributed along the slope. Buoyancy and fin control allow a quiet approach without costly, sustained pursuit. A feeding opportunity becomes a measured maneuver, matching limited energy supply. Returning before daylight reduces time exposed away from structural shelter. These repeated actions turn rugged geology into ecological infrastructure. Volcanic caves provide daytime refuge. Currents transport scents and prey, and depth controls temperature. If you remove one element, a seemingly broad ocean can become unsuitable. This dependence connects coelacanth history to modern fishing and habitat disturbance. Deep-set lines and other gear can capture animals that approach bait during nocturnal foraging. The cue is food odor. The action is approach, and the consequence may be lethal bycatch. Because reproduction is slow, even limited removals can matter for small local populations. Conservation assessments must work with incomplete counts and uneven observation. Some known groups have been repeatedly surveyed, while other plausible habitats remain scarcely examined. A lack of sightings may indicate absence, difficult terrain, limited effort, or all three. Climate-driven changes in temperature and oxygen could compress usable depth zones. Individuals may descend or relocate, but continuous suitable caves may not exist. Human curiosity also creates pressure when rare specimens become valuable. Responsible study, therefore, balances genetic sampling, observation, and protection of vulnerable animals. The coelacanth survived scientific invisibility through habitat and circumstance, not supernatural concealment. Conservation now depends on protecting verified refuges, while uncertainty still surrounds other possible habitats. Long-duration submersible surveys could test cave fidelity, nocturnal routes, and seasonal depth changes. Environmental DNA can screen water for genetic traces, though contamination and currents complicate interpretation. Genomic comparisons may reveal population isolation, past contractions, and limited exchange among regions. Future encounter evidence must preserve observations that might otherwise disappear into local memory. Each method addresses a different gap without turning a trace into a confirmed population. A DNA signal suggests presence, while direct imaging or a specimen establishes identity more firmly. Absence after one expedition cannot prove that a rugged slope holds no coelacanths. If you revisit the first harbor, the discovery now looks less like resurrection than connection. A fisher's catch entered a museum network able to recognize its evolutionary significance. The fish had persisted through deep time, while formal science had sampled only fragments of its range. Its caves reduced encounters, its night activity hid behavior, and its rarity delayed confirmation. Fossils supplied the old framework, but living bodies transformed that framework into ecology. Much remains unknown about birth, dispersal, hunting, and undiscovered habitat. Future evidence may extend the map, yet every claim must remain proportional to verification. The coelacanth's greatest concealment was the vast distance between oceanic existence and documented human knowledge.